Welcome, curious minds. Today, we're embarking on a deep dive into one of life's most incredible journeys, how animals, ourselves included, transform from, well, vulnerable youngsters into capable, thriving adults. Is this whole process of, profound physiological changes that shape pretty much everything an animal can do, from navigating the world to finding its next meal. Our insights for this deep dive come mainly from Chapter 4 of Animal Physiology by Hill, Wise, and Anderson. It's a really foundational text, just packed with compelling stuff.
Yeah, it really is. And our mission today, essentially, is to uncover the core concepts behind physiological development and this fascinating area called epigenetics. We'll look at the intricate mechanisms, how it all works under the hood. We'll compare strategies across different species, try to understand the adaptive significance, why these changes happen, and even touch on some of the clever experiments biologists use to figure this all out. You're going to get some great real-world examples that hopefully bring these complex ideas to life.
Okay, sounds great. Let's unpack this journey from infancy to adulthood then. And maybe we start with an animal that faces an almost, well, unbelievable challenge right from birth. Let's talk about the hooded seal. I mean, picture this. Born on unforgiving pack ice, these pups nurse for just 3 days. That's it. It's the shortest nursing period of any mammal on Earth. In that tiny window, they double their weight, going from maybe 20 kilograms to 40 kilograms, and then bam! They're on their own. They have to figure out how to find food in the vast, deep ocean. It's just an immediate high-stakes test.
It really is. And what's truly remarkable, physiologically speaking, is how their diving capabilities have to develop under such, well, extreme pressure. Adult hooded seals, for example, spend something like 90% of their time underwater. They're diving 3, maybe 4 times an hour, going to incredible depths, sometimes as deep as 1,000 meters. But those young, newly weaned seals, they're far, far more limited. And this limitation isn't just, an inconvenience. It's literally life or death for their survival and whether they'll ever reproduce.
Okay, so they're on their own super quickly. What are the absolute critical physiological things holding them back? What makes or breaks their survival right then?
Well, the primary limitation is their oxygen stores. Simple as that, almost. When animals dive, they're relying on oxygen stored in their lungs, sure, but also oxygen bound to hemoglobin in their blood, and really crucially, oxygen bound to myoglobin in their muscles. Myoglobin, that's the iron-containing protein that gives muscles their deep red color. More myoglobin, more oxygen storage in the muscle. And the data shows that a weanling hooded seal can only store about half as much oxygen per unit of body weight compared to an adult. But it's even more dramatic when you look at it per unit of tissue. The biggest bottleneck for these weanlings is their very low capacity to store O2 in their muscles with myoglobin. Adults have roughly 37 millimoles of O2 per kilogram of body weight bound to muscle myoglobin. Weanlings, only about 8 millimole, less than a quarter. And this isn't just because they have less muscle mass overall, it's also because the concentration of myoglobin within their muscle tissue is much lower. That concentration increases substantially as they mature, which actually makes their muscles visibly redder.
That's a staggering difference. I mean, less than a quarter. It really puts the physiological hurdles these young seals face into perspective. So what does that actually mean for their diving, for how they find food?
Well, precisely as you'd expect, their diving is severely restricted. While the weanlings also spend about 90% of their foraging time underwater, like the adults, most of their dives are really short, maybe 2 to 5 minutes long. Compare that to 5 to 25 minutes for adults. So weanlings end up doing about 14 short dives per hour versus just 4 longer dives for the adults. They're working harder for less reward, potentially. And the depths? Vastly different. Weanlings mostly stay above 20 meters. Adults, though, they regularly go 100 to 600 meters deep, and the record dives exceed 970 meters. So their hunting abilities are clearly fundamentally restricted by these physiological limits.
Wow. So the hooded seal example powerfully illustrates that development isn't just about getting bigger. It's a precise, almost life or death tuning of the internal physiology to meet these huge environmental demands right from day one.
Exactly, and this highlights a universal principle we call developmental physiology, or sometimes ontogeny. It's the study of how an organism develops from, well, conception right through to adulthood. The physiology of youngsters always differs significantly from adults. It changes gradually in these age-specific ways, and these changes are absolutely critical for an individual animal's ecological success and, ultimately, its evolutionary fitness.
Okay, so the hooded seal vividly showed us how physical systems like oxygen storage have to develop. But what about, the most complex organ, the brain? Let's maybe shift gears and talk about how this command center develops. And perhaps the most relatable example is, well, Us, humans. In humans, our brain matures incredibly rapidly. It reaches its full adult size by around 7 years old, which is amazing when you think that our general body tissues are still less than half developed at that point, and our reproductive organs have barely even started growing.
hmm And this rapid brain development, it raises a really important point about energy needs. Brain tissue has an incredibly high metabolic rate per gram. We're talking more than 10 times that of skeletal muscle. In adults, the brain accounts for about 20% of our total body metabolism, even though it's only about 2% of our body weight. But get this. In children aged 4 to 5, whose brains are nearly full-sized but whose bodies are still quite small, the brain accounts for roughly 50% of their total body metabolism.
50%.
50%. Half of their food intake goes just to fueling their brain. And that has pretty profound implications, right? It makes childhood starvation a particular threat to, arguably, our most crucial human tissue.
That's a really sobering thought, realizing where so much of a child's energy is actually going.
Hmm.
But okay, it's not just about size or metabolism. Brain performance also matures developmentally, often in these really fascinating ways. Like, think about birds like indigo buntings or garden warblers. They use stars to guide their nocturnal migrations. How does that complex skill even develop? They aren't just born knowing it, are they?
No, they're not. It's a truly elegant process. These young birds, during their very first summer, they don't have an innate star map. What they do is they observe the apparent rotation of the stars around the North Star, Polaris. Through this observation, their brains undergo a specific maturational change. They essentially develop a neural record, a kind of map, of those central stars and learn to associate them with North. And experiments in planetariums have just beautifully demonstrated this. If you raise young birds where they observe stars rotating around a different point in the simulated sky, like Betelgeuse or something, they will later use that point as north for navigation. Without that specific maturational period of observation and learning, they simply cannot determine compass directions from the stars. Their brain needs that environmental input to wire itself correctly.
That's incredible. So the brain is actively learning and changing based on experience right from the start. And it's not just brains, right? You mentioned thermoregulation earlier, just keeping warm. That ability also has to develop after birth.
That's absolutely right. While adult mammals and birds are, famous for their sophisticated homeothermy, maintaining that stable internal temperature, newborns are universally less effective at it. Take the white-footed mouse, a common example. Newborns cool down really quickly if they're exposed to cold environmental temperatures, even near freezing. They just don't have the machinery yet. But during their 3-week nestling period, a few key things happen. Their peak metabolic heat production dramatically increases. They grow fur for insulation, and their overall ability to thermoregulate as isolated individuals improves remarkably. By about 18 days old, a lone youngster can actually thermoregulate for several hours in freezing air, and that's when it starts its first excursions outside the nest, a critical step towards independence.
Okay, so development isn't just about getting bigger or smarter. It's really a fundamental restructuring of how the body works, right down to the cellular level, which must involve changes in specific molecules, specific proteins, and maybe even where those molecules are expressed in the body.
Precisely. Yeah, physiological development occurs at all levels, right down to the scale of individual proteins. The specific proteins expressed in any given tissue determine its structural properties, its metabolic properties, basically what it does. A really common pattern during tissue maturation is for key enzymes to be upregulated, meaning their production is increased in a very precise, stepwise fashion, all under genetic control. It's like a pre-programmed sequence. Think about liver enzymes in developing rats. Some key enzymes, like glycogen synthetase, which is critical for making liver glycogen stores, are abruptly upregulated just days before birth. Boom, the newborn has this vital ability immediately. Others, like phosphenol pyruvate carboxykinase, needed for making glucose, are upregulated right at birth. And then glucokinase, important for regulating blood glucose, comes online later in the nestling period. Each step is timed perfectly to meet the animal's changing needs.
That makes sense at that micro level, controlling which enzymes are active when. But what about a larger scale? Like you hinted, maybe tissues themselves change where they perform a function. Does the body actually shift the location of its physiological tasks as it grows?
Yes, absolutely. And a fantastic example of that functional shift comes from the killifish. It's a marine fish, so it lives in salt water and faces the constant challenge of becoming too salty. It needs to actively excrete chloride ions. In adult killifish, specialized cells called chloride cells located in the gills handle this vital process. But here's the kicker. Young killifish, under 8 days old, don't even have functional gills yet.
No gills. So how do they manage?
Well, biologists figured this out using a technique called immunocytochemistry. It's basically like using molecular tags that light up specific proteins. In this case, they look for a protein called NOT plus NASHK plus NASH-ATPase, a tiny pump that's super abundant in chloride cells. And they discovered that in these very young fish, the chloride cells aren't in the non-existent gills. They're found on the membrane of the yolk sac and scattered all over the skin surface. Wow.
Yeah. Then, as the fish mature and their gills start to develop, chloride cells begin appearing there. Eventually, in the adult fish, they are primarily located in the gills where we expect them. But for most individuals, because, sadly, death rates are high for young fish, the yolk sac and the skin are the main places doing this vital salt transport during that crucial early period. The function literally moves as the body develops.
That's incredible. The body literally shifts where it performs a vital function as it matures. Amazing. And all of this whole intricate developmental program, it's under tight genetic control, I assume. The genes are orchestrating this sequence.
You're absolutely right. Studies using advanced methods like DNA microarrays, which let scientists look at the activity of thousands of genes all at once, confirm this. They show that different sets of genes are transcribed or turned on at different specific times during development. And this leads directly to those sequential changes in protein expression and ultimately tissue function that we've been talking about. It's a highly orchestrated genetic dance. Okay, now let's maybe pivot slightly and explore something called phenotypic plasticity. It sounds a bit complex, but as I understand it, it's basically nature's way of creating multiple versions of the same genetic individual, right? Each sort of uniquely adapted to different environmental conditions it might encounter growing up.
That's a great way to put it. It's a truly remarkable phenomenon, and it really underscores that your adult characteristics aren't just rigidly predetermined by your genes. They're heavily influenced, shaped even, by the environment you experience during development. And we can see quite dramatic evidence of this even in human populations, looking back historically or comparing different groups today.
Right, like the example often cited is the average age of menarche, or first menstruation, in European girls. Back in the mid-19th century, it was typically around 16 to 17 years old, but by the late 20th century, that average had dropped significantly, down to around 13.
Exactly. And this shift wasn't due to rapid genetic evolution in just a century or so. It was almost certainly a direct consequence of vastly improved nutrition, public health, and medical care over that time. A more, let's say, benign childhood environment led to earlier reproductive maturity. We see similar effects in growth patterns too. Studies comparing children of Maya people in Guatemala, who often live under greater environmental stress, show they are, on average, about 5.5 centimeters shorter at each age than genetically similar Maya children whose families settled in the United States, where conditions are generally better.
And some of the historic data from the British Isles paints an even starker picture, doesn't it? Like factory children back in 1833 were apparently 16 to 20 centimeters shorter during their teenage years compared to modern children. And they were even shorter than aristocratic children living at the same time. Medieval villagers, going back further, were shorter still.
These cases strongly suggest that harsh environmental conditions, things like poor public health, chronic malnutrition, effectively prevented normal growth and maturation. They just couldn't reach their genetic potential. And this leads us to a really fascinating biological question. Are these environmental effects always just sort of damage? Are they forced changes caused by chemical or physical necessity leading to suboptimal development? Or could some of them actually be programmed responses, maybe even beneficial adaptations triggered by the environment?
That distinction between a programmed adaptation versus just damage or pathology, that seems really important. But how hard is it to tell them apart in the real world? How do scientists even begin to figure that out? Maybe some examples would help clarify.
It's a great question, and honestly, it's often quite challenging to definitively prove one way or the other. But let's look at some cases that seem clearer. Consider marine mollusks, like the flat periwinkle snail or the blue mussel. When these creatures grow up in the presence of crabs, even if they only detect the crabs through chemical cues in the water, they develop shells that are more than 10% thicker than snails or mussels growing without that cue.
Okay, thicker shells make sense for protection.
Exactly. This seems like a clear protective adaptation. It's very likely a genetically programmed response. Think about it. Why waste energy building a super thick shell if there are no predators around? But if predators are present, it's highly advantageous to invest in that defense. Natural selection would strongly favor individuals with this kind of flexible, plastic response.
Right, so that's a pretty clear example of an adaptive programmed response.
Yeah.
The environment signals danger, the genes have a program to respond beneficially. What about the other side, changes that seem more like they're simply forced, like damage?
Well, in contrast, think about studies looking at brain function in rats subjected to malnutrition during development. If young rats are fed a low-protein diet during that critical postnatal period, they often show impaired spatial learning ability later in life, even after their diet is improved back to normal.
So the deficit persists.
It often does, and this deficit is likely not an adaptation. It seems much more like a pathology, a situation where the brain simply could not develop normally because it lacked the essential building blocks, the proteins, due to that early malnutrition. It's a fundamental failure of development forced by the poor environment.
Okay, so some changes are clearly beneficial strategies, evolved responses, while others are really just damage caused by a tough environment preventing normal development. It does make you wonder, though, how many human traits we might think of as programmed could actually be forced, or maybe a complex mix.
Exactly, and you hit on a key point. In many real-world cases, especially in complex organisms like humans, it's incredibly difficult to definitively say. Some biologists, for instance, argue that slower growth or delayed menarche seen in human populations under harsh conditions could actually be a programmed adaptation.
How so?
Well, the idea might be there's an advantage to being smaller when food is perpetually scarce, or perhaps delaying reproduction until conditions improve. It's a strategy of waiting it out. Others, however, view these same outcomes simply as pathology's direct negative consequences of poor conditions preventing normal development. It highlights the complexity and the ongoing debate in developmental biology.
Okay, building on that idea of dramatic environmental influence, let's dive into some of the most striking examples of phenotypic plasticity, starting with something called polyphenic development.
Yes, this is where it gets really visually dramatic. Polyphenic development is where genetically identical individuals can develop into two or more distinct body forms, or morphs, entirely induced by environmental differences during their development. And these forms can be so incredibly different that, honestly, if you didn't know better, you might think they were entirely different species.
Like that western white butterfly example, where individuals developing in one season look quite different. Different wing patterns, different amounts of dark pigment, melanin, compared to those developing in another season.
Exactly. That's a classic case of seasonal polyphenism. It's typically cued by environmental factors like day length and temperature during larval development, and it plays a vital role in thermoregulation. Those darker wings seen in butterflies developing during cool seasons, like spring, help them absorb more solar radiation, warming them up faster, which is crucial for being able to fly when it's cool. In hot seasons, the lighter, different phenotypes presumably offer ways to avoid overheating. It's a really elegant, adaptive solution to predictable seasonal changes.
And maybe the most stunning and certainly impactful example of this is migratory locusts. We all have this image of them descending in massive, devastating swarms. But it's not simply that they are locusts that swarm. There's more to it.
That's absolutely right. Both of the major species of migratory locusts, Locusta migratoria and Schistocerca gregaria, exhibit polyphenic development. They have two main behavioral phenotypes, solitary and gregarious. And these forms differ dramatically, not just in their behavior, but also in their morphology, things like their body shape, their coloration. Yet crucially, there's essentially no genetic difference between a solitary locust and a gregarious one. They are the same genetically.
Wow. So what triggers the switch?
It's crowding, primarily.
Yeah.
Solitary locusts actively avoid each other. They're quite inconspicuous. But if environmental conditions, say dwindling food resources concentrating them in smaller areas, bring these solitary individuals into close contact, even for just a few hours, they begin to transform into the gregarious phenotype. They become highly social, attracted to each other, their appearance changes, they become more active, and they start to aggregate, eventually forming those massive migratory swarms. And we know this transformation is mediated by specific neurochemicals, biogenic amines like serotonin and dopamine, the very same kinds of chemical messengers that influence mood and behavior in us humans. It shows how fundamental these systems are.
So crowding itself acts as the environmental cue to flip this switch, changing their entire physiology and behavior. What's the adaptive advantage thought to be for this drastic change? Why swarm?
The leading hypothesis is that the fast-moving, highly voracious behavior of the swarm enables the locusts, as a group, to find and consume large quantities of food scattered over a very wide area. This becomes critical when local resources are depleted. So when solitary locusts become crowded, it acts as a signal. Hey, our population might be outstripping the local food supply. The transformation to the swarming gregarious phase allows them to basically enhance their collective food collection rate and move en masse to find new resources. It's a strategy for dealing with boom and bust population cycles.
That's an incredible level of responsiveness to an environmental cue, triggering such a profound shift.
It really is. And it's not just insects that do this kind of thing. Think about temperature-dependent sex determination in some reptiles, fish, lizards, turtles, where the incubation temperature of the egg actually dictates whether the offspring becomes male or female. That's polyphenism. Or Cope's gray tree frog tadpoles. If predators are present in their pond, detected chemically, they develop different behaviors and even different tail shapes, growing a deeper, often reddish tail that might act as a lure to distract a predator away from the head. Even tiny water fleas, Daphnia, can grow stronger, larger exoskeletons, sometimes with spines or helmets, if chemical cues from predators are present in the water. So while insects like locusts show perhaps the most dramatic forms, polyphenic development is surprisingly widespread across the animal kingdom.
Okay, this is where things get, I think, really mind-bending. Let's move into the world of epigenetics. It's a term that's generated a huge amount of buzz, especially in the last 20 years or so, and for very good reason. It seems to challenge some really fundamental ideas about genetics.
It absolutely does. Epigenetics basically refers to modifications of gene expression, how genes are turned on or off that happen without any change in the underlying DNA sequence itself. But, and this is key, these modifications can still be faithfully transmitted when cells divide, and sometimes even across generations. It's like adding another layer of information, another layer of control on top of the DNA sequence. Let me try to explain a core mechanism. DNA methylation. Imagine an environmental factor or maybe just normal developmental signals cause tiny chemical tags called methyl groups to attach to specific spots on a cell's DNA molecule. These methyl groups often act to sort of silence or at least reduce the expression of nearby genes. Think of it like putting a do not read sticky note on a specific paragraph in a book. Now, what's really crucial is that when that cell divides to make daughter cells, the cellular machinery often copies these methylation patterns along with the DNA. So the daughter cells inherit the same sticky notes, the same pattern of modified gene expression, even though their actual DNA sequence is identical to the original cells.
Okay, hang on. So it's like a memory, but it's not stored in the DNA letters themselves, but in these chemical tags attached to the DNA. And this memory gets passed down through cell divisions.
Exactly. That's a great way to think about it, a cellular memory. And this means that environmental influences that trigger epigenetic modifications early in an animal's development can potentially be passed on to all the cells that arise from those initial affected cell lineages, potentially affecting the whole organism throughout its life. And as you mentioned, some researchers argue pretty convincingly that these epigenetic marks can sometimes even be passed from parents to offspring across generations. This profoundly challenges the old dogma that genes are completely isolated from environmental influence between generations.
Right. So how does this really differ from the phenotypic plasticity we were just talking about, like the snails getting thicker shells? Because the environment is influencing the outcome in both cases. What's the core distinction?
That's a really good question, and the distinction is subtle but important. In programmed phenotypic plasticity, like those snails, the genes essentially contain the program that uses environmental information, like crab chemicals, to decide which phenotype to express, thick shell or thin shell. But the genes themselves aren't being physically lastingly modified by that environmental cue. In epigenetics, however, the environmental factor can directly mark the genes or the proteins packaging them in a way that's chemically stable and transmissible when the cell divides. This directly alters how those genes are expressed, often semi-permanently. The DNA sequence hasn't changed, but its accessibility or readability has, and that change gets copied. It's a more direct physical imprinting on the genome's function. And besides DNA methylation, there's another major mechanism, covalent modification of histone proteins.
Yeah.
Remember, DNA wraps around these spool-like proteins called histones inside the cell nucleus. Well, chemical changes to those histone spools, adding or removing different chemical tags, can also make the DNA wrapped around them more or less accessible to be read, thus altering gene expression. And these histone modifications can also be perpetuated during cell replication, contributing to that epigenetic memory.
Okay, this suddenly makes a lot of sense for how cells specialize in a multicellular organism,
Yeah.
right? I mean, all our cells, liver, muscle, brain, have the exact same set of genes, yet they look and function completely differently. Epigenetics must be absolutely key there.
It's absolutely foundational to it. Epigenetic marking is now understood to be a key mechanism driving tissue differentiation during development. Early on, the developmental program establishes different cell lineages, setting aside cells destined to become liver, muscle, neurons, etc., partly by creating unique, stable patterns of epigenetic marks on their DNA and histones. These marks then determine which specific sets of genes are expressed in each lineage, defining the cell's ultimate phenotype, its identity. And because these marks are faithfully copied during cell division, they ensure that the lineage's specific identity is maintained as the tissue grows and throughout the animal's life.
And there's growing evidence. You mentioned that epigenetics is also controlling some of those dramatic polyphenic developments, like caste differentiation in social insects. That seems like a perfect candidate.
Absolutely. It's a really active area of research. Take honeybees again. We know genetically identical female larvae can become either short-lived workers or long-lived reproductive queens, based entirely on whether they are fed royal jelly during development. Recent research has actually shown that if you experimentally manipulate DNA methylation levels in larvae that are otherwise destined to become workers, you can actually redirect their development, causing them to become queens instead.
Just by changing the methylation.
Just by changing the epigenetic marks. This epigenetic switch radically alters their physiology, their body size, their behavior, their lifespan. It's estimated that more than 20% of the genes in a developing female bee's brain are expressed differently depending on whether she becomes a queen or a worker, all driven by these epigenetic marks laid down in response to diet. Or look at the Florida carpenter ant. They have different worker castes, minors and majors with different jobs, especially foraging activity. Researchers were able to inject chemical agents that promote histoneacetylation, one of those histone modifications, directly into the brains of the larger major workers.
And what happened?
Astonishingly, these major workers started foraging far more actively, behaving much more like the smaller minor workers. This strongly suggests that epigenetic mechanisms, specifically histone modifications in this case, are controlling these caste-specific behaviors, effectively rewiring their activity patterns without changing their underlying genes.
This is just, oh, wow. It opens up so many possibilities. So the environment, diet, stress, social cues can induce these semi-permanent epigenetic changes that are then passed on not just within the individual cells throughout its life, but potentially, as you said, even across generations.
Yes. Now, the evidence for transgenerational epigenetic inheritance passing marks across generations via sperm or eggs is still more debated and complex, especially proving it happens robustly in natural environments. But there is compelling evidence in some model systems, including those water fleas, Daphnia, and possibly the migratory locusts we discussed. For the locusts, the current thinking is that the initial switch from solitary to gregarious, triggered by crowding, is indeed epigenetically controlled. What's even more intriguing is the idea that the epigenetic marks for gregariousness might then be partially transmitted to the offspring. This would mean that subsequent generations don't just experience crowding themselves, but they might also inherit some epigenetic marks that predispose them towards the gregarious phenotype, potentially intensifying and accelerating the swarm formation process across generations. It creates a kind of inherited memory of population density.
And this is where it circles back and gets incredibly relevant for humans too, doesn't it? Particularly thinking about the potential lifelong effects of early life stress or malnutrition. How does epigenetics tie into that?
Precisely. There's a rapidly accumulating body of evidence suggesting that early life adversity, things like stress, poor nutrition, exposure to toxins, can have profound, long-lasting physiological and behavioral effects decades later. And epigenetic marking is heavily implicated as a key biological mechanism mediating these effects. The idea is that these marks, induced early in development by adverse environmental factors, can be passed down through countless cell divisions and persist into adulthood years later, fundamentally altering adult physiology and maybe even disease risk. One of the most striking human studies looked at individuals who were conceived during the Dutch hunger winter famine, a period of severe starvation at the end of World War II in 1944-1945.
Right, I've heard of this study.
Yeah, it's quite famous now. More than 60 years after the famine, researchers found that these individuals, compared to their own same-sex siblings who were conceived outside the famine period, showed a statistically significant lower percentage of DNA methylation marks at specific locations in a relevant gene involved in metabolism, the IGF-2 gene.
Lower methylation. And since methylation often acts to reduce gene expression, does that suggest the famine effectively caused a change that might allow greater expression of certain genes in these individuals, even decades later?
That's exactly the implication. And this research isn't just, academic curiosity. These same individuals who were prenatally exposed to the famine show an unusually high prevalence of certain adult health problems, including obesity and coronary artery disease. It strongly suggests that the epigenetic changes wrought when they were just embryos in response to that extreme nutritional stress might be directly affecting their odds of developing serious health conditions decades later. It highlights this incredible biological link between the early life environment and long-term adult health outcomes.
And it's not just extreme events like famine, right? You mentioned earlier maternal care itself can have similar epigenetic effects. It seems like one of the most compelling lines of evidence.
It's truly one of the best documented examples studied extensively, particularly in lab rats. These studies, often using genetically very similar rats to control for genetic differences, show that the amount of licking and grooming LG, a mother rat, provides to her pups in the first week of life has lasting effects. Offspring of mothers who naturally provide low amounts of this care, the low LG mothers grow up to be adults who are significantly more fearful. They show greater hormonal responses to stress compared to offspring of mothers who provided high amounts of care, high LG mothers.
And they found epigenetic differences.
They did. Direct measurements showed significant differences in epigenetic marks, both DNA methylation and histone modifications in key genes within brain regions critical for regulating the stress response, like the hippocampus. It seems the level of care provided by the mother literally epigenetically programmed these lifelong differences in how their offspring respond to stress. It's a powerful example of environment shaping biology via epigenetics.
What an incredible journey we've been on, exploring this world of physiological development and epigenetics. From the immediate life-or-death physiological race against time in a newborn hooded seal, to the intricate navigational learning in a migrating bird's brain, and those dramatic, environment-driven transformations of a locust. We've seen just how dynamically animals adapt and mature.
We really have. We've uncovered some of the intricate mechanisms, things like oxygen storage limits, precise enzyme upregulation, the dynamic dance between genes and environment through phenotypic plasticity, and now these potentially semi-permanent imprints of epigenetics. It just profoundly underscores that an animal's phenotype, what it looks like, how it behaves, how its body works, isn't just rigidly predetermined by its genes. It's truly a dynamic product of this continuous interaction with its environment unfolding over its lifetime with consequences that can last that lifetime or maybe even ripple across generations.
So maybe the next time you observe any animal, young or old, consider that incredible developmental journey it undertook, and the profound ways its environment, especially early on, might have shaped its very being, right down to the fundamental expression of its genes.
And that perhaps raises a final thought for you, our listeners, to ponder. Given what we're learning about the potentially lifelong impact of early life experiences, particularly things like nutrition, stress, and maybe even care, through these epigenetic mechanisms, what might be the broader societal, maybe even ethical implications for human public health policies, especially concerning how we support nutrition and overall development in early childhood?
That's definitely some food for thought. We really hope this deep dive has given you some profound new perspectives on development and the environment.
Thank you, as always, for being part of the Last Minute Lecture family.